That's a question that probably many of us ask, and sometimes even I ask myself, because the definition is not very clear. But if you allow me to, I'll start with a brief analogy. And I'll talk about both genomic and genomic testing and then how they differ from each other. If you imagine how the X-rays were adopted back in the beginning of the 20th century to look inside the human body, and later on to determine how the tumor lives in the body and where it is. Before that point, doctors were tapping on the skin of a patient, trying to figure out what's inside. Then you had that X-ray that actually told you what's inside. And it was a groundbreaking discovery, but it wasn't that easy to adopt. It took many years of studying it, taking different parts, slices of the body to figure out where that blob is that you really want to address. Well, we've been tapping on the surface of the cell until recently. We didn't have an X-ray for the cell. So now we have the genomic and genetic testing as the X-ray of the cell. And that is, origin of cancer would become less and less leading in the way that cancer is determined and treated. The X-ray of the cell would become more and more important because it identifies how the DNA is folded, exists, and structured within the cell. And based on that DNA, you would know what kind of targeted therapy to prescribe to the patient. Now, if you think about the DNA in the cell, it probably would remind you of a big ball of yarn. And it would have beads woven into it, scattered, not too dense. Those are the genes. So the genes themselves are the pieces that really affect how everything works in the cell, but also the structure of the whole ball. Where is that bead? Is it on the surface or inside? And when we talk about genetic testing, now going back to the two types of testing, genetic and genomic, genetic really refers to the genes themselves, the beads. And very often when we have hereditary diseases, we carry them as mutations, or nowadays we refer to them as variants. And those are carried from generation to generation. They might have an effect on the patient. And usually they refer to small changes or sometimes bigger changes in how that bead has moved to a new place together with the pieces of DNA on both sides. But when we talk about genomic testing, then we look at the whole ball. Imagine you cut it with a knife on one side. You're not affecting only that one bead, but you're affecting the structure of the others. So instead of genetics, you're going to call it genomics to encounter for the whole field of that particular cell's DNA. So to summarize, genetic testing clearly is used nowadays for hereditary, usually limited mutations. Sometimes they can cover a whole chromosome, but not too complex with too many differences in them. When we talk about cancer, we're talking about genomic testing. And because cancerous cells are more or less like bacteria, they mutate with time, they adapt like bacteria towards different antibiotics. That's how cancer cells adapt against different targeted therapies. So you want to know the holistic look of the genome of the cell. And you're looking at the whole genome if you can, probably in the future, but now you're looking at a lot of different genes that are associated with cancer. And usually those genes are selected based on targeted therapies that exist currently on the market or development. The goal is really to link the targeted therapy to a particular genomic profile or a mutation in a cell.